Hello, everyone. I'm here with CLL Society. I'm Alan Skarbenik. I'm a CLL lymphoma doctor in the Avant Health Cancer Institute in North Carolina. I'm joined by Con Tam, who will introduce himself.
Hi, I'm Con Tam. I'm a CLL doctor from Peter MacCallum Cancer Centre and the University of Melbourne in Melbourne, Australia. And it's a real pleasure to be here today.
Thank you so much, Con. So we're going to talk about an abstract that was presented at ASH in 2021, for which you were the senior author. It's abstract 1549, selective Bruton tyrosine kinase inhibitor TG1701 as monotherapy and in combination with Ublituximab and Umbralisib, commonly known as U2, in patients with B-cell malignancies. So if you could go over the study, what the goals of the study were, and what your findings were, that would be great.
Sure. Thanks, Alan. So TG1701 is a next generation BTK inhibitor. It's very similar to acalabrutinib and zanubrutinib in that it is an improvement upon ibrutinib chemically. It's more specific than ibrutinib, but also it binds to the same spot irreversibly. So once again, it's in the same generation as acalabrutinib and zanubrutinib. So this study really reported on a follow-up on the phase 1 study using this BTK inhibitor. And to summarize, it behaved exactly as expected. It got good responses in the patients who were responsive to ibrutinib. There were no unusual side effects. It performed very similar to other next generation BTK inhibitors. What's a bit different about TG1701, of course, is that it's owned by TG Therapeutics, who also have Umbralisib, the PR3 kinase inhibitor, as well as the next generation anti-CD20, given the combination of U2, and because all of the assets are owned by the same company, it gave us the ability to combine a BTK inhibitor, a PR3 kinase inhibitor, and an anti-CD20 together as part of the phase 1 exploration in cohorts. And the long and the short of it is that the combination is actually really well tolerated. So adding TG1701 to U2 didn't substantially change the side effect profile of U2. And in those histologies, especially in follicular lymphoma, where we know that U2 is already very effective, we're seeing some very deep and encouraging responses. So at the moment, the big news is that we now have a feasibility for a triple combination. It's certainly very well tolerated. Is it better than the existing doublets? Well, the results are promising, but obviously we need further follow-up and more patients and probably some direct head-to-head studies to tell for sure.
Thank you so much. I think knowing toxicity here is key because we all had concerns with previous iterations of PI3 kinase inhibitors, in particular, with the side effects of diarrhea and concerns of combining with first generation BTK inhibitors. And it seems here in the study that the incidence of diarrhea was not more than what it was seen with U2 alone, right? By adding TG1701, is that correct?
Correct. So our impression is that it didn't really add to the side effects of U2. And of course, we're lucky to have Umbralisib, which is probably the one PI3 kinase inhibitor that's associated with the lowest risk of autoimmune colitis and pneumonitis. So I think the background of the PR3 kinase inhibitor really counts in addition to the BTK inhibitor.
That's great. And what are your thoughts on dual BCR inhibition here? You think it's going to catch those cells that would escape one of the enzymatic inhibitions, but targeting the same receptor pathway?
Yes, I think that is the hope, isn't it? But I guess the issue is that a single agent BCR inhibition with BTK inhibitor is working so well in the key diseases like CLL and Waldenstrom's that it's very hard to improve upon that. But I think things like mantle cell lymphoma and things that are less BTK responsive, those are the ones that I think will be a really promising target.
Perfect. Thank you so much. Much appreciated.
Thank you.